Have you ever wondered why the size of particles in pharmaceuticals matter? It might seem like a tiny detail, but this small factor plays an enormous role in the world of pharmaceuticals. In fact, it's quite the game changer in drug development and delivery. In the world of pharmaceuticals, particle size is more than a mere measurement. It's a critical aspect that influences how effective a drug can be. Imagine a drug as a key and our body as a lock. For the key to fit perfectly, its size and shape matter significantly, isn't it? Similarly, for a drug to work effectively, its particle size needs to be just right. The size of these particles can impact how quickly a drug is released into the body. Smaller particles tend to dissolve faster, which can lead to quicker absorption into the bloodstream. So, if you've ever wondered why some medications kick in faster than others, you now have your answer. But it's not just about speed. Particle size also affects the distribution of the drug within our bodies. Larger particles may get stuck in one part of the body, while smaller particles can travel further. This can influence how effectively a drug targets a particular area of the body. Beyond efficacy and distribution, particle size also plays a role in the physical stability of the drug. It can influence aspects like the shelf life of the medication, its appearance, and even its taste. So, the next time you notice a bitter taste in your medication, remember, it could be due to the particle size. In the fascinating world of pharmaceuticals, it's clear that size does indeed matter. From affecting how quickly and effectively a drug works to influencing its physical properties, particle size plays a pivotal role. Now that you have a glimpse of why particle size matters, let's delve deeper into its significance. Stay tuned as we venture further into this fascinating topic and understand the methods of measuring particle size and the significance of size separation in pharmaceuticals. The size of particles in pharmaceuticals plays a pivotal role in determining many factors. You may wonder, how so? Let me explain. Firstly, particle size is key in enhancing drug solubility. The smaller the particle size, the larger the surface area in contact with the solvent. This higher surface area allows for increased dissolution rate, leading to improved solubility. Imagine a sugar cube dissolving in a cup of tea. If you break that cube into smaller pieces, it dissolves much more quickly, right? It's the same principle here. Next, particle size also controls the rate of drug release. In many drug formulations, the active pharmaceutical ingredient, or API, is embedded within particles. The size of these particles determines how quickly the drug is released into the body. Smaller particles often mean a faster release, while larger particles result in a slower, more controlled release. Think of it as the difference between a sprint and a marathon. Then, we have the stability of formulations. Here's an interesting fact. The particle size of a drug can actually influence its stability. Smaller particles have a larger surface area exposed to the environment, which can increase the likelihood of chemical reactions, potentially affecting the stability of the drug. So, in some cases, larger particles may be preferred to maintain the stability of the formulation. Finally, let's talk about patient compliance. Particle size can significantly affect the texture and taste of a medicine, especially in oral dosage forms. Smaller particles can lead to a smoother texture and better taste, enhancing patient compliance. Imagine trying to swallow a tablet that feels like sandpaper versus one that's smooth as silk. Which would you prefer? In a nutshell, from enhancing drug solubility and controlling the rate of drug release, to influencing the stability of formulations and improving patient compliance, the size of particles plays a crucial role in the success of a drug formulation. Clearly, the size of particles is not something to be overlooked in the pharmaceutical industry. Measuring particle size is no less crucial than understanding its importance. So let's dive into the primary methods used for measuring particle size. First up is microscopy, the most direct method of particle size measurement. Here, individual particles are observed under a microscope, and their dimensions are measured directly. This method is highly accurate for small particles and allows for detailed morphological studies. However, it tends to be time-consuming and requires significant skill and expertise. Next, we have laser diffraction, a method that measures particle size based on the scattering pattern produced when a laser beam passes through a sample. This method provides rapid and accurate measurement of a wide range of particle sizes. It's advantageous because it can analyze a large number of particles simultaneously, providing a comprehensive size distribution profile. However, it's worth noting that this method assumes particles are spherical, which may not always be the case, potentially leading to inaccuracies. Last but not least, we have sieving, a classic and straightforward method of particle size measurement. Here, particles are separated based on their ability to pass through a series of sieves with varying mesh sizes. It's simple, cost-effective, and ideal for larger particles. However, it's less effective for very small particles, and it doesn't provide information about particle shape or specific dimensions. In summary, each method has its pros and cons. Microscopy offers high accuracy and detailed information, but is time-consuming and requires expertise. Laser diffraction is rapid and provides a comprehensive size distribution profile but assumes spherical particles. Sieving is simple and cost-effective but is less effective for small particles and doesn't provide shape information. Choosing the right method depends on the specific needs of the analysis, the type of particles, and the available resources. By understanding these methods and their limitations, pharmaceutical professionals can make informed decisions and accurately determine particle size. With these methods, pharmaceutical professionals can accurately determine particle size. Scene script: size separation is another critical aspect when it comes to particles in pharmaceuticals. Let's dive right into it. Size separation plays a pivotal role in the formulation of drugs. It assists in creating a well-balanced, uniform mixture of multiple components. Imagine a cake mix. If the sugar granules were larger than the flour particles, they wouldn't blend well, resulting in an uneven taste. Similarly, in pharmaceuticals, if the drug particles are not uniformly sized, they might not distribute evenly, leading to inconsistent dosages. Furthermore, size separation ensures the uniformity of dosage. In the world of pharmaceuticals, precision is key. Each dose must deliver the exact amount of active ingredients to be effective and safe. The particle size affects how quickly a drug is absorbed into the body, its solubility, and its bioavailability. By separating particles based on size, we can ensure that each dosage will behave predictably and consistently, offering the same therapeutic benefits. Now, onto the quality of pharmaceutical products. Size separation is instrumental in improving this as well. Larger particles often lead to a gritty texture, while smaller particles can result in a smoother product. This can impact not only the user's experience but also the stability of the product. Size separation can help to reduce these issues, leading to a higher quality product that is more appealing to users. Lastly, let's touch upon the techniques used for size separation. Two commonly used methods are sieving and sedimentation. Sieving separates particles based on their size using a series of screens with different sized holes. Sedimentation, on the other hand, separates particles based on their size, shape, and density in a fluid. Each method has its own advantages and is chosen based on the specific requirements of the drug being produced. Size separation, thus, aids in maintaining the integrity and effectiveness of pharmaceutical products. It truly is a fascinating and vital process in the pharmaceutical industry. Particle size and size separation in pharmaceuticals are not just technical jargons, but integral to the drug development process. As we've journeyed through this topic, we've unpacked the significance of these concepts, their measurement methods, and their implications in the pharmaceutical industry. We started by understanding how particle size in pharmaceuticals is not just about tiny granules. It's about how these little units interact with each other and with their environment, influencing the drug's dissolution, bioavailability, stability, and even the manufacturing process. It's a small detail with a big impact, making it crucial to get it right. Next, we explored various measurement methods of particle size. From sieving and microscopy to more sophisticated techniques like laser diffraction and dynamic light scattering, each one has its unique advantages and challenges. The choice of method is often dictated by the nature of the drug, its intended use, and the level of accuracy required. It's a fine balance of science and art, precision and pragmatism. Then we delved into the significance of size separation. This concept is so vital because it helps to ensure uniformity and consistency. By separating particles based on size, we can control the drug's properties more effectively, resulting in a product that is safer and more reliable for patients. Size separation techniques such as sedimentation, centrifugation, or chromatography are commonly used, each with its own set of benefits and constraints. So, as we wrap up, remember that particle size and size separation are more than just a part of the pharmaceutical jargon. They are fundamental aspects of drug development, with far-reaching implications on the efficacy, safety, and quality of the drugs we use every day. Understanding these concepts can help in the production of more effective and safe drugs. Subscribe for more such enlightening content.